Okay, so today I'm going to talk about resting state fMRI and using the resting state fMRI data to look at everything except neuronal activity. And I'm also going to talk about a mysterious signal that we're trying to understand now. So my perspective is that the brain is just a bag of blood, or it's a series of tubes like the internet. And the blood, just enters through the arteries, goes through all the little vessels, and comes out in the veins at the end. Blood flow has a constant component, which is just the bulk flow. And then it has a pulsatil component, which is due to cardiac pulsatility. Both components of this signal are interesting, and they both contain information. And the neurons probably use the blood for something too, but that's not relevant. They're just there to hold the blood vessels up. But more seriously, neuronal activity alters cerebral blood flow in detectable ways, or fMRI would not be nearly as popular as it is. But that's only some fraction of the signal. Up to 50% of the signal in the gray matter can be due to other hemodynamic factors, such as the things I just described. What people did in the first days of NMR is they just sort of they dealt with it, and they said, well, we'll just average it away because it's noise, right? Noise averages away, the signal adds up coherently. And that's great, and that worked well for a while people were doing tasks related to fMRI. But then when they started doing resting state fMRI, you don't have a task. And then the question becomes, how do you make sure that what you're averaging is what you want and not some other signal? Especially the problem with the low frequency signals is they're right in band for the resting state connectivity. So rather than just try to average and hope that they go away, what you need to do is understand them, then you can model them, then you can intelligently remove them, or you can use them for something else. Okay, so I'm going to talk about two frequency bands, if I have time, otherwise I'll just talk about one. The first is low frequency oscillation band, and that's what roughly goes from 0.09 to 0.15 hertz. The people define the boundaries of this band differently, but that's what we use in our lab. And then there's the cardiac band that goes from about 0.7 hertz to 3 hertz when you include harmonics. So there are prominent blood effects in both bands, but they're completely different in terms of their mechanism, their spatial distribution, and their propagation velocity. So you need to really consider them separately. Okay, so this is a picture of, I'm going to say it's a typical fMRI voxel, but it's not typical. It's done with a very short TR, 400 milliseconds. And it was chosen to be sort of a pathological amount of cardiac contamination, which is over there on the right. Usually it's not this bad, but all of these things are in your signal in every voxel in the brain. The fact that we do fMRI often with a TR of 2 seconds or 1 second doesn't mean these components aren't here. It just means that you're not properly representing them. And one thing that people always used to say is, well, there's no point in going faster than a 2-second TR because the rise time of fMRI is 2 seconds, and there's no signal change fast in that regime. But the noise changes faster than that. And if you don't properly digitize your noise, then you can't remove it properly, unless you play some tricks. And this is just shows you what various TRs are going to get you in terms of aliasing. With TR of 4 seconds, you get everything up to the primary of the cardiac signal. You're not going to get the harmonics, but as you go down into this band in the blue, as long as you're better than about 800 milliseconds, you can fully catch the respiratory band. So that's fully digitized. You can use it. You can motion correct without aliasing, and you can just apply a high pass filter. If you're doing fMRI, everything you care about is in this green band. Now, unfortunately, and so that means you just need a TR of 3 seconds to see that. But all of this other stuff is just going to fold back into there if you haven't dealt with it in some way. And unfortunately, one of the noise signals is also in that band. It just lives there. So no spectral filter is going to remove it. You need to do something else. Okay, so LFO band noise. In 2010, Yunji and I were looking for stuff to do, and we were trying to understand why the near signal from a frontal voxel correlated really strongly with our fMRI data. I mean, for the experiment we were doing, we wanted it to correlate somewhat, but it was correlating all over the brain, and that didn't make any sense. So this led to basically my entire research program, and we're looking at a propagating signal that's moving with the blood. It took a while to establish this, but I think we've pretty much firmly decided that. And we developed methods to isolate this signal and fully characterize it. And then once you do that, you can fit it and you can either remove it from your data if you care about doing functional connectivity, or you can use it for something else. And we find that doing something else with it is more interesting. So here's this is one of the experiments we did. This is not the experiment we actually discovered this on, but this has better pictures. So we were doing a simultaneous bold and NIRS experiment on the motor cortex. Finger tapping experiment. Everybody knows what this looks like. We were able to do tomographic reconstruction of our NIRS signal. And we thought, this is great. We can overlay the bold signal and the NIRS signal, and we can see the areas of activation where they're the same, where they're different. We can get a sense of how the bold signal is individually affected by changes in oxy and deoxyhemoglobin. Then we had this bright idea that what if we took the NIRS signal, which was digitized at 25 hertz, and just used that as a probe to look at to our GLM to record to get the bold activation in the bold data. And our rationale for that is, everybody's always talking about the hemodynamic lag and what is the hemodynamic response function, and you have to assume something. If we just measure it with NIRS, then we're going to get a great response. And that kind of works. I mean, if you see on the bottom here, this is just the standard block time course involved with the standard hemodynamic response. And this is the bold response in the motor cortex that you would get. When you take the NIRS signal and filter it to the appropriate band, you get this, which, okay, that looks pretty much the same. I'm getting the bold activation, but it doesn't look better than that. And in fact, in many ways, it looks substantially worse. And the reason for that is that we've got all this junk all over the brain. And we didn't like that because that was not what we expected. And moreover, NIRS can't see more than about 2 or 3 centimeters into the brain. So how could the NIRS probe, which was placed directly over this activation, see any of this stuff at the base of the brain? That didn't make any sense to us. And in fact, when you put the NIRS probe on the other side, this didn't get any better. It looked pretty much the same, except that the activation that you were looking for went away. So the fact that it was detecting activation all over the brain, as I said, didn't make any sense to us. So we had to figure out why we were getting this. So one of the things that we saw is it's very similar when we used a probe from the task and the non-task region. So what we're looking at is probably, is a widespread effect. It's also unlikely to be a machine artifact because NIRS and bold are completely different. Different machines, completely different noise mechanisms. The idea that they would share an artifact that would be synchronous and it would cause this type of correlation just didn't make any sense to us. So what ties them together is what you're actually looking at, your sample. You got physiology under there. So at first we thought, well, maybe it's a resting state network that people don't know about. And we're actually seeing connections between this part of the brain and every other part of the brain. It wasn't that. First of all, it had no coherent structure. It didn't resemble any of the resting state networks that you see with any other technique. So we thought it's probably some sort of global signal carried in the blood. And the reason we thought this is that if you look at a raw NIRS signal before you filter it, the biggest part of the signal is not task, and it's not cardiac, although cardiac is quite large. It's actually these huge low-frequency oscillations, anti-correlated to the oxy and deoxyhemoglobin, implying that they're oxygenation fluctuations. And that's actually the largest amount of the signal power that we usually get. So we figured, well, that's there. Maybe that's what we're seeing. Maybe that's tying into our bold somehow. So we thought that if we were seeing these, then we should be able to test it. If it is a systemic blood oxygenation change that we're seeing in the NIRS, then it should move because oxy and deoxyhemoglobin are carried in blood. They have to physically move from one part of the brain to the other, and they have to go along the vessels to do that. So if we were to change the alignment between the NIRS signal and the bold signal, then the positions that we see these activations occurring in should move. And so keep in mind that we can consider an individual slice in an fMRI scan to be a single time point. It's acquired over 30 milliseconds, but that's a very short time compared to the frequencies of the oscillations. So in terms of that, the fMRI image, it's static during that time. So when we did that, we basically just took the time delay between the NIRS probe and the bold and we just changed it from minus 15 seconds to plus 15 seconds. We sort of scanned through it. And then it looked a lot more orderly. And what really stood out to us is that at the longest time delay between the nearest probe and the fMRI data, we saw it coming up here in the superior sagittal sinus. We didn't know this was a superior sagittal sinus at the time because we didn't know anything about the vasculature. But then we saw this kind of cool picture of in one of our time of flight angiograms. It's like, oh, wow, there's this big honking vein. So it's probably something blood related. So we decided to study this in isolation rather than as an accidental confound in a task-related scan. And I say that like, oh, yes, this is our research plan. What it meant was we were so excited about having one subject where we saw this, we sent it to NeuroImage, and they're like, no, do a real experiment and get back to us. So we eliminated the task because that was the first thing that people said, oh, you're getting a task response. We looked at 6 subjects, but most importantly, we came up with an acronym to describe our technique, because that's branding is what this is all about, really. So we called it Riptide, Regressor Interpolation at Progressive Time Delays, and that's where the name Riptide comes from. So it turns out that worked. And when you do this in 6 subjects and you average the data over 6 healthy controls, you get what we saw before, except you see it in more parts of the brain and it's more orderly. And it really looks very much like the vasculature. Blood seems to appear in the middle of the brain, sort of propagates out, and then it ends up going through the draining vessels. And if you render this in 3D, you can actually see the vessels that go up to the superior sagittal sinus and then go back. Now, when I say this is a global signal, I mean it's a global signal. So we saw it in the brain, and we saw it all over the brain, and we caught it with a probe on the scalp. But the results that you just saw, the probe was on the fingertip, because the fingertip correlates very well with the brain. And in fact, the fingertip correlates with the frontal lobe. Cheek, right here, the zygomatic arch, correlates with the frontal lobe. The fingers and the toes correlate. We went crazy. We were like putting nurse probes everywhere. Finger and yeah, so we got excited and we were looking all of it. And we actually have a sort of side business doing looking at peripheral vascular physiology, because you can actually look at the time delays between the legs and between the arms and the earlobes, and you can learn things about peripheral artery disease. So, the engineer in me says, well, what can you use this for? Well, I mean, if you've got 50% of the variance in the volt signal accounted for by this, it's got to be useful for something, right? So if we can find it reliably and figure out a way to really fit that in every voxel, then we can split the volt signal into 2 components, the component that stays put and the component that moves around. Now, if you're looking, which of those is signal and which of those is noise is completely a matter of perspective. So if the moving component is signal and neuronal activation is noise, which is my perspective, we can learn all sorts of things about circulation because we can track blood passing through the brain. We can make maps of RCBV or something very much like RCBV because it's actually weighted by blood oxygenation, as Yunji was talking about. And we can see, most important, we can see alterations in circulation due to pathology. This was our theory. We figured, stroke, atherosclerosis, moya, all of those things would affect circulation, would affect when blood arrived at various parts of the brain. Mostly what happened is we got so tired of looking at 27-year-old healthy graduate students. They all looked exactly the same. We figured that, if this is going to be useful, there's got to be some difference in the signal somewhere. So anyway, if you could do this with the resting state scan alone, this simple scan, you get this for free. You don't have to do ASL. You don't have to add time to your exam. You don't have to have a special technique, and you can use data that you already have. Okay, so here's an example. I'm going to cheat a little here, and I'm going to show you some data that we got with working with Manus. This is from moya. This is not using endogenous noise, but it does show the pattern of evolution of blood as it flows through the brain. So keep that in mind. This is better than any data we would normally get in a single subject. This is looking at one of his endarterectomy patients. On the first panel, you can see the time delays before surgery, and then you can see it 6 months after revascularization. and then 22 months after revascularization. It's important to look down at there at the bottom, at the bottom scale. The end of this, the largest time lag at the end of this moving is 140 seconds. So the first time we started talking to people about this, they're like, well, there are no time delays that long in the brain. And it's like, well, how do you know that? It's like, well, ASL, you never see things that long. It's like, no, you wouldn't, right? Because the tag can't last that long. I was very happy at one ISMRM. I actually saw somebody who was doing CT angiography where they used logarithmically spaced time points. So rather than, if the problem is if you go out to 140 seconds at the time resolution you usually do for CT, you cook your subject, and that's not good. So she had the brilliant idea that, I'll take them, 0 time, 0.1 seconds, 0.2, 0.4, 0.8, and just keep taking them, take the same number of steps, but over a longer period. And she actually saw things like this around the number of drug. She saw very long time delays. And that was the first time that I think people had ever seen that. This is just another rendering of that. This is showing the maximum time delay rendered on the surface of the brain. You can see that function is restored very well. And people use this clinically using just endogenous signals currently. The one I'm most familiar with is Ahmed Khalil, who's using this clinically at the Charité University and hospital in Berlin. They're making decisions as to whether people have ischemic or hemorrhagic strokes using a 2.5-minute resting state scan that's reconstructed on the scanner, and they can put delay maps up, and they can make decisions based on that. So if the moving component is noise, which is the perspective of most of, like when I go to OHBM, this is all I want to hear about. You can generate and regress out voxel-specific confounds, confound regressors from your fMRI analysis. And that models the in-band low-frequency oscillation noise. And it does so in a way that is a lot better than existing methods. So we did a study in 2016 where we looked at 100 subjects from the HCP, young adult, and we did global signal regression, which is sort of the state of the art. And we did our method, and we did no regression. And what we found is that our method removed the largest amount of variance from the brain in a very spatially heterogeneous way, but it removes the noise from where it is, but it removes it far more effectively than global signal regression. And the reason it does that is the global signal is sort of a smeared version of this low frequency signal I'm talking about. If you take a signal with a very, the highest frequency in this signal is 0.15 hertz, right? So it's about a 6-second period. So it takes about 6 seconds for blood to get through the brain. So if you take a bunch of signals with a highest frequency component of 6 seconds and you add it up in sort of a Gaussian histogram, it's going to look like a lot like the original signal. It's just going to be a little blurry. So what happens when you're doing global signal regression is you get perfect denoising in the very small number of voxels where you're exactly aligned in time. And then every other voxel, you're regressing out the right signal at the wrong time. And what that leads to is a negative copy of a delayed signal being imposed on your data. And that's where the spurious negative correlations in fMRI come from. You're doing a global signal regression. And you see these purple bars doing global signal regression. You're getting, these are, the red is, before you've done regression, you have correlation between everything in the brain, right? Because everything has blood in it. Then when you do global signal regression, you get a lot of regions. These are purple regions. These are regions that are known not to be neuronally connected to the regions we were looking at, and yet they have a negative correlation. And when you do rapid tide, they're actually pretty much uncorrelated. You don't make the spurious negative correlation. Anyway, so we argue that this is a better way to denoise your data. So over the years, we've made a lot of methodological improvements. As I said, we use cross-correlation analysis rather than FSL-GLM, which goes orders of magnitude faster for the processing. We found that since we asserted that this global mean regressor is everywhere in the brain, you got to be able to find it in the brain somewhere, right? So we developed an iterative bootstrapping method to start from the global signal and refine it until we figured out what this moving signal looked like. That means you don't have to have an external NIRS probe. That's great because most existing data doesn't have that. And it's it's just one more thing that you have to take care of if you have another instrument. Yunji has validated this against dynamic susceptibility contrast imaging. We spent a lot of time over the last 13 years sort of establishing that this was a blood signal. People kept saying, "Oh, it's not. It's something else." Well, what would it be? It moves exactly as fast as gadolinium does in the brain, so that was helpful in making our case. The fact that you can find it in the arteries before you actually hit any neurons helped with the argument that it's not a neuronal signal. I mean, the fact that you can find it in the finger should have done that, except people were saying, well, it comes out of the brain and goes to the finger. So anyway, we've released this package. I talked about it yesterday. So it makes it pretty much turnkey. If you just want to denoise data, it'll just do that out of the box. You really don't have to tweak any knobs. It'll pretty much do that. If you want to do delay mapping, that's a more complicated problem because you can't solve things in the quick and dirty way. But I mean, for most data, it's fine. For CPR data, it's great because that's an enormous signal. If you're using endogenous noise that comes with whatever characteristics it comes with, that can be twitchy, but I can talk about that later. Because now I want to talk about the mysterious LFO signal behavior because Alex wanted to talk about it. We recently saw something weird in one of our experiments. Some of my collaborators, despite my advice, still try to look at neurons. So I worked with a group that was doing an analysis of nicotine Q reactivity. Basically had this Q reactivity experiment, which elicited a very strong response to a task. And what she also found, she wanted to look at, does this affect long-term insular striatal connectivity? Does it potentiate over time? Like by the time you could see this thing 5 times, are you like, clawing at the inside of the magnet to have a cigarette? So we actually saw that. We saw the connectivity. We started with the resting state, and then we did the task 5 times. And we found that the connectivity between the regions of interest increased over the entire time course. I thought, great. And then we thought, wait a minute, nothing ever works the way we expect it to. And we said, it turns out the connectivity increased between areas that we knew were unrelated to the task. And in fact, the connectivity increased all over the brain. Well, that's not good. So we ran rapid time denoising on the data, and the effect went away, along with the result which we wanted. But that, neither here nor there. It seems that physiological LFO signal is not stable. It grows in time as you're lying there in the magnet. So we thought, well, okay, well, maybe we just screwed up. It's happened before. Maybe let's look at something where people did better data collection. So we used the HCP young adult data set because we use that for everything. We actually have a paper that we've been sitting on for coming up on 7 years, where we looked at every single resting state scan in the young adult database. That's 4,036 scans. So this is the average delay map on the left and the average correlation map on the right. So we know what to expect from this. You get a pretty robust signal when you average that many points together. So we figured the data is good enough. There's enough of it. Let's break the data up into 4 blocks, right? 4 segments. And sure enough, the signal correlation strength gets bigger over time. And not, this sort of global way, in a very structured way that matches the pattern that we saw in the previous slide. We're seeing that pattern on the right, except it's getting stronger and stronger over time. And if you go on to the second scan, they always start. HCP collected its data. The first resting state volumes were collected. They did a right-left phase encode and then a left-right phase encode. Right, left, left, right. And then second data did the other way, left, right, right, left. Which means by looking at the names, which one happened first. So if you look at the first scan, the signal starts very low here and grows. When we go to the second scan, it doesn't go back to where it was before. It's still high. It doesn't stay where it was, but it dips a little, and then it continues to grow. And you can see that summarized here on the graph. This is overall average conductivity over the whole brain. So we thought, that's kind of weird. So we thought, can we look at this a little more directly? So we took the normalized global mean signal from the entire brain for every subject, for every run. And so they're all normalized to percent bold activation they start at 1. And then we just calculated what is the variance of the time course at every time point over all subjects. And we found that the global mean signal in the low frequency oscillation band grows over the entire length of this 15-minute scan. It's maybe starting to roll over near the end. It's a little hard to say. And then when you do the second scan of the day, it goes up like that again. And this is replicated in the opposite order on the second day. The first scan started low and went high. The second scan started a little higher than the first one did and continued to go high. And we found that rapid tide, for the most part, really removed that. This signal strength pretty much doubles over that time period. And after you do rapid tide, it goes up by maybe 10 to 15%. It's worth noting that if you look at the frequency band outside of the LFO region, you're not seeing that same sort of growth. We're not getting a twofold increase in the signal power. One thing we did find is that people probably don't throw out nearly enough time points at the beginning of the scan. I mean, I look at that and I say you should throw out the first 100 time points, but that's interesting. T1 decay seems to last a lot longer than you would think it does. So we thought, well, maybe it's, and by the way, other people have seen this before. Jeanette Bisterbach at Oxford has seen this, did do this in a single scan stuff. They looked at a change in connectivity over the entire brain. But I don't think they really attributed it to a systemic signal like this. I think they thought it was related to arousal, which may be indirectly. So we thought we better check on some other data sets. So we tried another one. This is a data set that we found in Open Neuro, and this has a resting state data and a sleep data set. We see the same thing. And so one of the arguments about this is that perhaps it's arousal. But then it seems a little odd that if you look at the low frequency variance over the sleep study, it's growing over the entire time. These people are nominally asleep for the entire scan. So I'm not sure what that means. And again, the high frequency signal, which is shown in red, doesn't seem to change at all over the course of the scan. So what is it? I mean, it can't increase without limit, but it certainly seems to increase for 15 minutes. Persistent across an hour-long scan, it seems to just keep bumping up. It resets a little every time you change scans. Doesn't seem to be due to heart rate changes, because the heart rate we checked using PhysioPy, and the heart rate seems to come down in about 2 or 3 minutes and stabilize and stay there. It could be related to respiration, but not to respiratory rate, again, characterized with PhysioPy. It might have to do with arousal, and we have some evidence looking at pupillometry data in yet another data set that it is inversely correlated with arousal, but I don't understand how that matches with the sleep data set. Whatever it is, we can make it go away, and that's good. So if you, and the engineer in me says that it doesn't matter, as long as you can make it go away, it's not as important that you know what it is. But people are weird that way. They're like, if you see this big signal, what could it be? So I was hoping that this group might have some ideas about that. I can't think of a better group to ask. Okay, so moving on to cardiac band noise. I do have time for that.
Yeah.
So can we do something similar for cardiac pulsatile signal? Now, people who were here yesterday already saw some of this, and if you get bored, you want to leave, that's fine. But simultaneous multi-slice imaging takes data a lot faster than TR. I mean, any fMRI data does it. Scanners typically run at about 10 to 15 slices per second, or slice stacks per second, because that's as fast as the gradients can go without dumping too much heat. But multiband imaging allows people to take a stack of slices for each time the gradient beeps. So the number of slices you can take is not as directly tied to how much heat you're generating in the gradient set. That means you can sample, as I say, stacks of slices at a very high rate. And if you make some assumptions and you make an observation, you can use this to digitize things faster than you want to be able to. Here's just a little image of the cardiac signal pulsing away. This is, I think, 150 millisecond TR, or maybe it was 400 millisecond TR signal. You can sample the cardiac rate directly in the whole brain using multiband acquisition. But most data sets don't run that fast. So why do I care this much about getting a cardiac waveform? Well, I mean, it's an important noise confound, but the most important driving factor for this is in 2018, I saw this amazingly cool paper by Henning Voss. It's called Hypersampling with Analytic Phase Projection. And it was just the simplest idea, but it was brilliant. Basically, if you have the cardiac waveform over the course of an fMRI scan, and and what you can do, use a Hilbert transform to calculate the cardiac phase at any time during the scan. And then, since you know when every slice was acquired in the brain relative to the beginning of the TR, then you can calculate the phase of every single time point that's recorded over the course of your experiment. And if you know the cardiac phase for every time point, you can then rebin it into you can make a single cardiac cycle by rebinning the data, and that is it's like a Cine acquisition. That's what they did. With doing heart imaging that way. It's not exactly the same, but it's pretty close, and it's really neat. And I wanted to do that. And none of my data had cardiac waveforms. So I thought, well, first let me see if I can implement this. So this is a human connectome young adult data set. They record simultaneous plethysmogram data. And you can make this really cool pulsatility map. And I thought, well, that's great. I want to do that. Because I thought, this will help me find vessels, and maybe I can use it to artificially generate tops. And I had great plans for this. I thought, well, I should try it in another data set and see if it works. This is the myconnectome data set. This is Russ Poldrack 2 times a week for a year, a few years. I mean, that is real dedication. Anyway, and you can generate a map, and this is a much less fancy scan. I mean, we thought maybe the HCP data, well, it's multiband factor of 8, TR of 720 milliseconds. Maybe you just need to have super extreme acquisition parameters. This is much milder acquisition, multiband factor of 4 and a TR of 1.1 seconds. So if we could do it with this, then it should be possible. Anyway, so if you don't have a recorded waveform, the point of the second part of HAPI was to pull it out of the data. And the fact that, again, like with rapid type, if this is a pervasive signal, it's everywhere, you've got to be able to find it, right? It's just lurking there, waiting for you. So we thought we made one observation and 3 assumptions. The first observation is that while any given voxel is only sampled once per TR, you're sampling something at an adequate rate to catch the cardiac frequency. So you're typically running at 10 to 20 hertz. That should be fine. Okay, great. The first assumption is that the cardiac signal is sufficiently coherent within a given slice that you can just average all the voxels and say that's a time point, whatever that means. That's not actually true, but it's mostly true, and it turns out to be true-ish, and then true-ish enough that you can just pretend it's true and go and move blindly forward. And in fact, if you restrict to the blood vessels, it becomes truer. The second assumption is that the effect of the cardiac signal across slices is similar enough that you can find a way to intelligently combine the data sets from every stack of slices. And this is where having a high multiband factor helps, because the higher your multiband factor, then the more slices you collect at a single time. And if you do that, if you're spanning the brain each time with 8 slices, each of those stacks of 8 is going to look kind of like every other part of the stack. Cover kind of the same amount of tissue. It turns out that even doesn't have to be that true, because TR of 4, multiband factor of 4 works fine too. So you need to find a way of intelligently merging the time courses you get from each of these. And the third is that we know that the cardiac signal is periodic, pseudo-periodic. That helps a lot, right? Because that gives you some symmetry that you can use to clean the signal up once you get something to begin with. Okay, so the HCP example, Human Connectome project has 72 slices acquired in 720 milliseconds. That means you have 9 stacks of 8 slices collected every 12.5 milliseconds, which is wicked fast, and that we can do that. So we want to know, can we combine them? The answer, of course, is yes, or I wouldn't be standing here talking about it. So I showed this yesterday. What you do is you take these average time courses for each stack of slices, and you just graph them out for every single one of the stacks of slices. And then since you know when each stack of slices was collected relative to the TR, the beginning of the TR, you time shift them by that amount because you know that's when they actually happened. So if you put them all on a graph at their appropriate time points and then combine them at the appropriate time point, you do end up with a coherent time course. And it works remarkably well. Okay, so fine print, there's a lot of bookkeeping associated with this. I find, okay. And so obviously you got to take care of all this. Software does it for you, freely available in the RapidType package. We also took this original signal and we refined it. And we can make something that looks an awful lot like the plethysmogram. And from that, you use a CNN filter to do this. We get a very clean signal, which gives us a lot of spectral components that we're missing. Why do you do this? Don't people record simultaneous plethysmograms? Well, the answer is not always. I mean, we don't typically, or we didn't. And even when you do, like in the HCP data, about 12% of the plethysmograms are bad. I don't know, people had nail polish or the thing fell off or whatever. So if you wanted to analyze that 12% of the data, you're out of luck. But now you have a second way to generate this data. So you can get it even if you didn't have it or if there was a failure. And it's nice to have that some version of the cardiac waveform because then you can make this call out. Or do beat-to-beat heart rate detail and calculate HRV and all of that, calculate emotional response and all of that. I tried to do noise removal, using these signals. I never got a very good result, but Giulio Ferrazzi and Daniela Maranazzo came up with something called Who Cares, which starts from happy and then builds these alias time courses. And it does a very effective job at removing noise from fMRI data. So again, this lets you intelligently get to the part of the signal that you actually care about. So the conclusion is there's an awful lot of variance in resting state fMRI that has nothing to do with neuronal activity, but it isn't noise. It's a superposition of multiple spatial or temporal pattern signals that carry information about various human parameters. With a little thought and a lot of processing power, you can sort of lure it out of the data and then do whatever you want with it. Even better, you can do it with other people's data. So you don't even have to take data anymore. You can just retrospectively mine things. You look at the OASIS data set, you want to look at Alzheimer's. If they didn't do the experiment you wanted, you don't care, right? Because you got it. Anyway, thank you for your attention. None of this would have been possible without all of the collaborators that I work with. I've had the advantage of working with some very, very good people over the years, and they've done most of the cool work.